Today we'll be giving you a tutorial on basic genetics. Now, genetics is the transmission of traits from one generation to another, and these traits are going to be passed on from parents to offspring via chromosomes. Here you have a picture of a chromosome that's going to be found in the nucleus, and these are only visible when the cell is dividing, so during mitosis and meiosis. Within the chromosome is going to be housed DNA, deoxyribonucleic acid. And this is the chemical that will actually contain the code, the genetic code for the transmission of traits from parents to offspring. Here we have a picture of a human karyotype showing the chromosomes that exist. There are 22 pairs of what we call autosomes. Now, autosomes are non-sex chromosomes that are going to be found in the nucleus. And as you can see, each pair of chromosomes has its own size and shape. These are each called homologous chromosomes. There are 22 pairs of autosomes and then one pair of sex chromosomes. Now, the sex chromosomes will be XX in the female and then XY in the male. The female is homologous. As you can see, they have the same size and shape. The male, the Y chromosome, is going to be smaller than the X. So this is a non-homologous pair. When we say that chromosomes are homologous, as you saw in the previous slide, they have the same size, shape, and chemical composition. What does that mean, chemical composition? That means that the genes are going to be organized in the same sequence on the pair. So genes that are adjacent to each other are controlling the same trait. Another word for gene is allele. We're going to be using letters to represent genes or alleles. For example, in humans, eye color can be determined by an allele that we will represent with a capital B, and that's going to represent the color brown, or an allele that's going to be represented by a lowercase b, and that's going to represent the color blue. Now, if the allele is represented with a capital letter, that means it's dominant. If it's represented by a lowercase letter, it's recessive. And that means that once a dominant gene is present, or an allele that is dominant is present, the recessive one cannot be expressed. You will only be able to see the dominant gene. So let's say this is a pair of homologous chromosomes. And for each pair of homologous chromosomes, we received one from our mother and one from our father. So the genes that are going to be found on this homologous pair are going to be controlling the same traits, but they don't have to have the same information. For example, let's say this allele here is for eye color, and the mother gave the child a dominant gene for brown, but the father gave the child a recessive gene for blue eye color. So in this case, this child would have brown eyes. And in this case, the child would have the genotype of capital B, lowercase b. The genotypes are always expressed with letters. We can't see the genes. This is what's in the chromosomes, but we will be able to see the phenotypic expression. So in this case, capital B, lowercase b would give you brown eye color. So the phenotype is always expressed with a word describing the trait that you're talking about. Now, in this case, If the organism has a dominant and a recessive allele, then the organism is heterozygous for this trait. Another word that means heterozygous is hybrid. What's another way you can have brown eyes? You could also be homozygous dominant. Homozygous means the two letters are going to be the same. And also another word refers to homozygous is pure. So you can be pure brown, and in order to be blue, you have to be pure. That's the only way you will see the recessive trait. It has to be in the homozygous condition. So genotypes, always letters. Brown could be heterozygous, as we have here, or homozygous. This is also going to give you brown. The genotype for blue has to be homozygous recessive, and the phenotype for this condition now, two lowercase b's, will be blue. It's important that you understand the meaning of all these terms because you will be using them as we do problems in genetics. And remember, we have 23 pairs of homologous chromosomes. The exception is the male has 22 pairs because the sex chromosomes X and Y are not homologous. Here you have a picture of some traits that are inherited in humans. The one that we're going to be concentrating on right now is the shape of the earlobe. You can have unattached earlobes where the lobe will loop down. This is a dominant trait, so we're going to be representing it with a capital E for unattached. So a person with unattached earlobes can be homozygous dominant or heterozygous. Attached earlobes, there's no loop. It comes straight to the side of the face, and this condition is recessive. So attached earlobes can only be expressed in the homozygous recessive condition. Let's look at a cross now between two parents that are both heterozygous for unattached earlobes. Okay, so if we're crossing two individuals that are both heterozygous for unattached earlobes, the first thing you have to do is make a key. Decide what letters you want to use to represent the traits. As we had in the previous screen, capital E will represent the dominant trait, unattached. Lowercase e will represent the recessive trait, attached. So the first thing you do is declare the parental phenotypes, the P1 generation, the first parental generation phenotypes. What do they look like? As I said in the problem, they're both heterozygous unattached. Both of them are the same. Now, what is the P1 genotype? Use the letters from your key to represent the genotypes. Heterozygous unattached, capital E lowercase e, crossed with capital E lowercase e. They're both the same. So in this case, the female and the male both have the same genotypes. The next step is to indicate what gametes are going to be formed by each of these individuals. Well, remember, these letters are representing alleles or genes that are carried on the homologous pair of chromosomes. And if you remembered when we talked about mitosis and meiosis, especially meiosis right now for the formation of the gametes, the homologous chromosomes separated so that some gametes would have the capital E and some gametes would have the lowercase e. And that's what we're working with here. Both of these individuals are going to be producing the same type of gametes, capital E and lowercase e. Now you're going to take that information and put it into your Punnett square. So we're going to make a box, and we're going to put one parent on the top and one parent on the side. And it doesn't make any difference which one you put where, because the results will be the same inside. So we're going to put the female eggs up here, and for the male, the sperm are going to come along the side. And what you're going to represent inside of each of these squares are the possible combination of the sperm and the egg. So if this sperm fertilizes this egg, you're going to bring them down and across. And here's what you will produce. Individual that's homozygous dominant. If this sperm fertilizes this egg, you will have a heterozygous condition. If this sperm fertilizes this egg, again, a heterozygous condition, and always put the capital letter first. And the last box, if this sperm fertilizes this egg, you will have a homozygous recessive condition. Once you've completed your Punnett square, now you're going to look at the results and indicate how many different genotypes do you see. Well, we have homozygous dominant. How many? 25%, or 1/4. Heterozygous, 50%, half. And homozygous recessive, again, 25%. This is the genotypic ratio of the cross that we just produced. Another way you can represent this is 1 to 2 to 1. Now, what's the phenotypic ratio? What are you going to see? How many are attached? How many are unattached? Anybody that has the dominant gene is going to show the dominant trait. So that means 75% will be unattached, and only 25% will be attached. This can also be represented in a 3 to 1 ratio. Anytime you have two parents that are both heterozygous, you will always have a 3 to 1 ratio in the phenotype. This is called a monohybrid cross, where both parents are hybrid, and you're talking about just one trait, monohybrid. You will always reproduce this 3 to 1 ratio in the phenotype. Here is another example of a Punnett square, but this time the parents, one is going to be heterozygous, and the other one is, here's the heterozygous parent, and the other one is homozygous recessive. And as you can see, the results of this cross will give you, in the genotypic ratio, 50-50. The genotypic ratio, you have 50% showing the heterozygous condition and 50% showing the homozygous recessive condition. This can also be represented 1 to 1. And the phenotypic ratio is given to you here. So 50-50, 50% unattached, and 50% attached earlobes. So you would follow the same basic format in completing any other basic genetics problems that you will have in Chapter 2.